Hello everyone, myself Hanuman Gaikwad, a graduate student from North Dakota State University. Today's topic for my presentation is the mechanical response of forced activated integrin using molecular dynamics. As we know how cancer is affecting human life, so it's very important to know each and every element which affects the cancer progression. So here we considered integrin as one of the elements for our study. So what are these integrins? Integrins are the transmembrane proteins which couples the extracellular matrix to the cell cytoskeleton and facilitate the cell migration, proliferation, and differentiation. These integrins are composed of alpha and beta subunits. In initial stage, these integrins are in deactivated form. So once the ligand from the extracellular side or taline from the intracellular side couples the integrin, then integrin get activated and the bending to unbending transformation takes place. In metastasis, this activated integrin helps the tumor cells to penetrate the blood vessel as well as squeezed out from the blood vessels during the intravasation and extravasation process. Therefore, the mechanotransduction of the integrin during the cell adhesion has to be studied. So to investigate the integrin mechanotransduction, integrin alpha v beta 3 4-MMX protein structure is selected from the protein data bank for the simulation. The simulation has been divided into 3 models: protein models, protein methionine models, and protein fibronectin models. In initial stage, all these models were energy minimized and brought to the temperature and body temperature and pressure condition. And then these models are equilibrated for the same body temperature and pressure condition for 1 nanosecond. Then equilibrated model is used for the force pulling, where the tail end of the integrin is fixed and head end of the integrin is pulled. Here, all the tensile deformation for all the integrin models are plotted, where we observed same trend, same stress regime for all the integrin models. At the same time, we observe the activation of the integrin during the equilibration affects the stiffness of the integrin in initial pulling. Forced activated integrin structure then used for the bending and torsion stress. During the bending test, the tail end of the integrin is fixed and at the head end the force is applied. Here for protein fibronectin model, less force is required for same amount of deformation as compared with other integrin models. As like bending test, in the torsion test also the tail end is fixed and at the free end the torsion is applied. Here for all the models, angular deformation increases as the angular acceleration increases. To conclude the study, in the presence of methionine and fibronectin, we observe the activation of the integrin without the application of force, and this activation of the integrin affects the stiffness of the integrin. The resulted graph from all the tests then used for the plotting or calculating the mechanical properties of the integrin. Finally, I would like to thank my advisor Dr. Dinesh Katti, co-advisor Kalpana Katti, and all the funding indices for my consistent support. I would like to thank CCAST NDSU and Civil Environmental and Construction Engineering Department for their computational support and facility. Thank you.